Hi guys, happy snow day. I can't afford to miss a lecture, so I'm going to record this one for you guys. Luckily, a lot of this stuff is um, different applications of the stuff that you did in 1610. I do think it's really important that you watch this video because we are going to use this stuff throughout the rest of the semester. All I'm going to do is finish up the lecture notes on metabolism, and which is really just about proteins right here. And then I will get started and I'll do the easy parts of cell transport on this video, and leave the harder parts for when I see you on Wednesday. Okay, so um, we did most of this. Well, we did all of this in the 10 o'clock class. If you didn't get all of it in the 8:30 class because you guys are a couple minutes behind, please go back and watch the last few minutes of the last video. Um, in addition, there's a really, really good interactive about lipids and lipid digestion right here. If you click right here, it goes to Howard Hughes Medical Institute. Click Launch Interactive right here. And you can go through how the body uses fat. It's really, really good. It's actually really interesting. Talks about insulin, talks about weight gain, talks about glycogen, talks about a whole bunch of stuff that's just useful for you as a medical care professional, um, and also for this class. Okay, so let's finish up this real quick. So basically what we were doing is we were looking through this in multiple different ways. The first thing, of course, that we did was talk about how the body breaks down carbohydrates. We used glucose as an example to produce ATP, right? And the fact that this portion is anaerobic and everything after that is aerobic. Then the next thing we did was talked about how you break down fats. And that most fatty acids, although not glycerol, which is part sugar, part fat, most fatty acids actually skip the glycolysis portion and go straight through, and they actually generate more energy per gram than carbs. And then the last thing that we have to do is talk about how you can use proteins um, to generate ATP via catabolic processes. And this we're not going to do in a ton of detail, but proteins generate about as much ATP per gram as carbs do, and fats generate the most. But of course, you can use proteins for other things. So let's talk about this. All right. So if we look at, let me make this a little bigger. If we look at protein and amino acid metabolism, so the first thing that has to happen for you to use proteins for catabolic processes, actually it has to happen for any, first thing that has to happen for absorption is that you have to break down a protein, which is large and typically folded in maybe tertiary or quaternary folds. You have to break it down to a peptide, which is just a string of amino acids. And then you have to take down, and by the way, this is done primarily with the enzyme pepsin, and there are pepsins produced from multiple different places, including the pancreas. And then take the peptide, which is just the chain of amino acids, and then break it down to the individual amino acids. So take a protein and break it down to individual amino acids. This process um, is prior to absorption. And so what you have to do is you have to break it down to, these are the 20 amino acids. You have mechanisms of absorption that we'll talk about for each of these. And then you can take the amino acids and they're going to enter the bloodstream. And then you can use them either for catabolic processes, like breaking down, this would be a catabolic process, use an amino acid. Or if there are extras, you can use them for anabolic processes. But before you do that, if you're going to use them for catabolic processes, what you're going to have to do is you're going to have to take the amino acid, and I don't know if you guys remember this, but you've probably seen it before. All amino acids have this little nitrogen containing group on them. amino acids have this on them, and what you're going to have to do, except for this one, don't worry about that one. And what you're going to have to do is you're going to have to use oxygen, so it's oxidative, and you are going to have to do oxidative deamination. And what that actually does is removes the amine group right here. And only after you have done that can you burn those via catabolic processes for ATP. But important thing about that, this is ammonia, NH3, which is toxic. You can get rid of it directly, or you take most ammonia and you actually send it to the liver to detoxify or lower the toxicity, I should say, from ammonia to urea. And then, of course, you have to get rid of urea and other nitrogenous wastes via the kidneys. So, take home message here: protein catabolism requires liver function and kidney function to generate with to deal with this byproduct that you've generated. So, you have to excrete this urea and also some ammonia into the urine in the kidneys. So let's say that you had plenty of ATP in order to maintain homeostasis, and you had plenty of amino acids for all of your basic functionality. If there were amino acids and ATP left over, then you could use the extra amino acids for protein synthesis. That's an anabolic process, but it doesn't get dibs. For instance, if you were deficient in either amino acids or in ATP, you would not commit to this anabolic process. You would use the proteins that you ingested for a catabolic process. In addition, last little tidbit about just amino acids is that every organism, living organism, has the ability to make amino acids, but we don't all make the same ones. So there are 9 of the 20 amino acids that humans do not make enough of in order to not eat them, generally. I mean, you make some of these, and some of them you don't really make in any appreciable amounts at all. And these are the things that you're constantly seeing advertised as the essential amino acids that you have to ingest in order to have enough of them to use for instance, anabolic processes. And the only other little tidbit to tell you guys about this is that there are some other essential nutrients that are not these big macromolecules that you need in, for instance, gram quantities, like proteins and like carbohydrates and fats. And those are, again, back to the whole idea of vitamins and minerals. The only difference between a vitamin and a mineral is really whether it's inorganic, meaning not carbon-based, not made by a living thing, or organic, carbon-based, made by a living thing. And just for your own health and your own edification, the 2 big categories of vitamins are water-soluble and lipid-soluble. Most vitamins are water-soluble. The lipid-soluble ones are A, D, E, and K, ADEC. And water-soluble vitamins, if you take in excess, for instance, if you took a mega dose of vitamin C, like my mom used to give me when I was a kid because she thought that that was the way to make you healthy. Literature does not support that. What would happen to that 3,000 milligrams of vitamin C that I took? Well, it's water soluble, and if I were otherwise healthy, I would just pee out the excess because you clear water soluble excesses and also toxins, but this isn't necessarily toxic, through your kidneys. Now, don't get me wrong, I'm not saying that you cannot become toxic with a water soluble vitamin. You can overwhelm your kidney's ability to get rid of excess of anything. So don't try it. But lipid soluble vitamins are even more likely to accumulate because, of course, our kidneys are not effective at cleansing lipid soluble things from the blood plasma. So we tend to store them, and we store them in our fat tissue. And those can and do become toxic in high doses. And when they're sold over the counter, and some of them are and some of them aren't, like you can't buy vitamin K over the counter, you can buy D, and you can buy E. I don't think you can buy an A. I don't actually remember if you can buy A. I know that A is sold in larger quantities or used in larger quantities as a medication. But when they are sold, They are usually sold in smaller concentrations, like they're called international units or sometimes microgram quantities. Okay, and that's it for that set of notes. We just had a tiny bit more to do. Okay, and then the next set of notes is really just revisiting cell transport, but trying to make sure that you approach it from a physiological perspective instead of just a 1610 perspective. Because what we are going to end up doing with cell membrane transport is you're going to have to be able to predict, instead of me telling you how something will be transported, you're going to have to gain the ability to predict how it will be transported, because of course you're going to run into things that are not exactly like the example you've used. So what I want to do is I want to go through this Roman numeral number 2, and if you're not confident with this stuff, you're going to have to go back and review. Roman numeral number 2 starts with just the mechanisms of moving substances across the plasma membrane. So let's revisit our idea of the plasma membrane. So you guys know this, but basically it's a phospholipid bilayer. The phosphate heads are hydrophilic, and so they face toward the fluids. The phosphate tails are hydrophobic, and they face the internal portion. And then you quite often will have proteins that are considered integral proteins. This one goes all the way through the cell membrane, so it's both integral and transmembrane. This one does as well. Okay, so if, for instance, something could not pass through the lipid portion of the cell membrane, then you might be able to get it through a transmembrane protein. But how do you predict whether it would be able to go through the lipid portion or not? And how to, whether it would need a channel or whether it would need a carrier. So that's what we're going to talk about right now. So the plasma membrane is basically a boundary between the intracellular fluid and the extracellular fluid, but it's not an impermeable boundary. Of course, you've heard a million times that it's selectively permeable, or sometimes they call it differential permeability. So what is it that determines the permeability of a substance? It's really the relative constant nature of the plasma membrane, which is primarily lipids but with embedded proteins. And then the chemistry of the solute that you're trying to move across. I like this little animation. It's linked for you in your notes. This little animation that kind of gives us a way to look at these as we're going through. Okay, so let's just start with the basics. So how do you predict? So if you were trying to predict whether something can go across the cell membrane, first look at its size. Generally speaking, small things traverse the plasma membrane more easily than large things, meaning that this would be more likely to be able to go across the plasma membrane than, for instance, an entire cell. Okay, so size does matter. So small chemicals are more likely to be able to go across the plasma membrane. But then the next thing you have to consider is the concept of electrical charge or polarity. So charged particles and charged things like ions, like potassium, sodium, chloride ions, they're polar, and so they tend to be hydrophilic and lipophobic. So they're not lipid soluble, and again, the plasma membrane is primarily lipids. So if you have something that is strongly charged like an ion, generally speaking, it's not going to be able to get through the lipid portion of the plasma membrane. So that matters, the charge. Again, that's because they tend to be hydrophilic and lipophobic. Okay, now, can you get them across? Well, yes, you can get them across, but generally speaking, this is the time in which you are going to need to use a transmembrane protein, a protein that goes all the way across. So for instance, if I tried to do potassium, and it doesn't show this here, but this potassium is an ion and this sodium is an ion. So if I tried to get potassium across, is it going to pass here or is it going to pass here? And why? The why is important. So notice that potassium can go, and by the way, it was going down its concentration gradient through this ion channel, okay? It is not going to go through the lipid portion. And that is not because it's large, because potassium is a single atom right, with an electron removed because it's a positive ion. Okay, so you can get those across channel proteins. Here's a channel protein. Okay, um, now we'll come back to channel proteins in a few minutes, and then the other concept to deal with is just basically what about the solubility of the solute that you're trying to get across? Well, what matters here is primarily we already know that ions are hydrophobic or lipid soluble, but what about other things? Because other things are not necessarily strongly charged like ions, but they do have a hydrophilic or hydrophobic nature. So lipid soluble substances, if it is lipid soluble, it is going to be nonpolar. And of course, since the cell membrane is mostly lipid, then it is going to be soluble in the cell membrane, and it's not going to need to use a transmembrane protein. A really good example of that is like oxygen, lipid soluble. Steroid hormones, those are the fatty hormones, and some fatty acids. How would they get across the cell membrane? Would they, for instance, need to use one of these transmembrane proteins, or would they not? And by the way, you can read this, or you can hide the text when you don't want to see that. You're probably not going to read it on my screen. You can read it on yours. So how would oxygen pass? And this is oxygen, our little friend here, right here, just two oxygen molecule, or two oxygen atoms making one oxygen gas molecule. So it goes straight through. And contrast that with potassium that must use an ion channel. Hopefully that visual is helping you, especially for you super visual people. Okay, what about substances that aren't necessarily lipid soluble, but they're still kind of tiny? Very small water soluble substances like carbon dioxide, urea, and a few others seem to be able to cheat the whole solubility question because they're small enough. So how does carbon dioxide, which is generated inside your cells, and this is what it looks like right here, how does carbon dioxide traverse the cell membrane? Notice that it seems to be small enough, despite its solubility issues, that it can actually diffuse straight out of the cell. And you can mess with these on your own, by the way. Okay, so, but again, I want you to be able to think about why, okay? What if it's a mid-sized water-soluble molecule, meaning that it's not lipid-soluble and it's getting a little bigger? So is it going to be able to sneak straight across the cell membrane, or how will it get across the cell membrane? Like small sugar, like glucose or fructose, or an amino acid that you need to, for instance, get into the cell for protein synthesis, or getting a sugar into the cell for catabolic processes like cellular respiration. How do you get that across? Well, let's look. So glucose is water soluble and it's relatively large, so it's not going to go through the lipid portion of the cell membrane. And interestingly, glucose is actually too big to go through channels. So the next thing that we will see is that it can go through a carrier protein. And the carrier protein, I just want you to look at the difference, and I'll show you another picture of it too. So watch this ion channel. Notice that the ion channel doesn't really change anything about its conformation when, for instance, this potassium ion is going through. But watch the glucose carrier called a GLUT transporter. Again, notice that it has to move. And an even better look at it is this from your textbook. I realize it doesn't move, but it does show you, and this is important, that carriers, unlike channels, this is a channel, this is a carrier, have a shape-specific binding site for the solute that they are going to carry. And what they do is they actually bind to it. That changes their conformation to allow you to dump it, okay? That changes their conformation to allow you to use it again, okay? So keep that in mind because I'm going to ask you guys a couple questions to think about in just a second. Okay, so carrier protein. What about something that's really, really big? So it's water soluble. It's not tiny like carbon dioxide. It's not mid-sized like a sugar, a small sugar, or an amino acid. Is there any way to get a big water-soluble molecule, like a whole protein that was just made inside the cell, out of the cell? Of course there is, because you must have the ability to do transport of, for instance, let's say that this was, this was a pancreatic beta cell that just made insulin. Obviously, the insulin won't do anybody a damn bit of good if it stays inside the cell. I must have a mechanism of getting it across. An enzyme is also a protein. So how do you get the enzyme out? What you do with the enzyme is you get it out by what we call exocytosis, or if it's big and you want to get it in, you get it in by endocytosis. This is what we call vesicular transport, and it's energetically expensive and it's pretty slow. Okay, so now let's go back to this for just a second and say, okay, I am trying to transport a substance across the cell membrane. How do I predict whether I would do straight diffusion, like oxygen does and like carbon dioxide does, diffusion through a channel, like for instance potassium does, okay, diffusion through a carrier, like glucose does, or vesicular transport, like protein secretion. How do you predict which one you are going to do? Well, generally speaking, here's your prediction. You are going to use the cheapest, fastest way that you possibly can. So what we need to think about is which of these is going to be fastest, and then the ones that actually will use ATP. I didn't talk about active transport, by the way, but we will get to it in just a second. So active transport actually costs you money as well. We're going to learn it in detail on Wednesday when I see you guys, or cost you ATP. So I want you to think conceptually about whether straight diffusion through the cell membrane, like oxygen does, is going to be faster or slower than diffusion through a channel, like potassium does. So think about it this way. If you can do straight diffusion through the cell membrane, there's very little limitation on where you could diffuse. You have all of the surface area to diffuse, right? Wherever there are lipids. Whereas if you have to do diffusion through a channel, it's going to limit the number of places that you can actually diffuse. With that in mind, I hope that you can figure out that diffusion straight through the cell membrane is going to be faster than diffusion through this ion channel. And then what about comparing this ion channel where potassium diffuses to this glucose carrier? So I want you to think about which of those is going to be fastest. They are both free, no ATP required, as long as the concentration gradient favors them going in that direction. So if you look at this, this is a channel and you can diffuse straight through it down your concentration gradient. And so does the channel actually have to change to bind to the solute, change conformation, and then change conformation again? No, it doesn't. It's just basically like an open door, and just whatever you can fit through it will go through it. So hopefully that you can see that that would be faster than, for instance, this, where you have to bind, right? Wait for it to bind, change conformation, dump it out, reset the initial conformation to use it again. So that is actually going to be slower. So in order of how quick these are, straight diffusion is the fastest. Diffusion through an ion channel is the second fastest. This is called facilitated diffusion when it goes through a carrier. That's the third fastest. Okay, and then then we get into the ones that actually cost ATP. And so this one's a pump. And the only difference between a carrier, like this glucose transport, and a pump is that a pump is going to push something the opposite direction of its concentration gradient. So when you use a pump, what would make this, what this is the sodium potassium pump, what would make this actually a pump is that if sodium is high here, what I'm trying to do is pump it so that it's higher. So you're actually moving it against its concentration gradient. So it requires a carrier plus ATP. So of the ones that we've done so far, this is the slowest one. And then even slower than that is this process right here called exocytosis or vesicular transport. So one more time before we leave this concept, and we'll talk about the water one in just a second. So fastest, straight diffusion, and we're going to use this as we go through. Second fastest, diffusion through an ion channel because there are fewer places to diffuse. Okay. Third fastest, diffusion through a carrier because it has to bind, change conformation, bind, change conformation. Slower and now starting to cost us ATP is this protein pump, and the very, very slowest would be endo and exocytosis. So that is what you are going to use as your predictive thing. And note to self, when you guys start telling me as I'm asking you questions in class how something is going to move across the cell membrane, I'm going to be super annoying and ask you always why. Like, why will glucose not use a channel? Well, glucose, we will see over and over again, is too large, so it can't do straight diffusion because it's water soluble, not lipid soluble. Can't go through an ion channel because it's too large, so it has to do the third one. Okay, and then you don't have to move to the fourth one. So one more time with the visual, and then I will move on to the next concept, and this is straight diffusion, right? Down its concentration gradient. This is also straight diffusion through the cell membrane. If that won't work because this is not charged, it's not because of the size, sorry, it is because of the charge. It's not because of the size, it's because it's strongly charged. So why can't potassium do straight diffusion? Because it's charged. So it has to use an ion channel, okay? But then I want to move something else. I want to say, okay, I'm going to move glucose now. Why can't glucose go straight through the cell membrane? Because it is water soluble, not lipid soluble. Why can't it use an ion channel? Too big. So next best thing is to use a carrier, okay? And then when I'm moving sodium and potassium against their concentration gradient, they have to use a pump because it's going the wrong direction. It's basically like getting water to go uphill, up its concentration gradient instead of down its concentration gradient. And then last but not least, how about I just made some insulin inside the cell. It is water soluble, won't fit through a channel, won't fit through any of these carriers. How do I get it out still? I'm going to use exocytosis, which is a type of vesicular transport. So you need to get used to those kinds of thought processes. Okay, so cheapest, fastest way first. So now let's just look at this figure one. You should be able to sort of figure out which one's which. And I think I did all the channels. So there are a couple of different kinds of channels. Where did I put them? I put them down here. Okay, we'll do that in a minute then. I'll put them down there. I rearranged the notes and forgot that I did. Okay, so now let's just talk about the kinds of transport that exist. So you guys have done this before. Simple diffusion is just moving molecules, solutes, whatever you're moving, could be gases, could be ions, anything, from an area of high concentration to areas of low concentration. What's really happening in a situation like that is simply that if I actually just looked at potassium, now let's not use potassium, let's use oxygen. If I looked at oxygen and I start out with oxygen, what is true is there's more oxygen out here than there is in here, and oxygen's just going to move down its concentration gradient until the concentration is equalized on both sides. It's not directed. Each individual little oxygen molecule does not know which direction it's going. They're basically just smashing into one another until they're equal, until they're smashing into one another about the same over here as they are over there. It's called Brownian motion. It's not directed, but it's going to be passive, doesn't require ATP, but it does depend on a concentration gradient. And so the net movement is from high solute concentration to low solute concentration, but there's no directed movement of any individual particle. They just smash around until they all have about the same amount of room. And so distance, the diffusion, basically the simple diffusion, really only works well over microscopic distances, meaning that, okay, my lungs take in oxygen and there's a high concentration of oxygen in the tissues around my alveoli, and my little toe needs oxygen, but is diffusion going to be an effective transport mechanism between my lungs and my little toe? No, it's too far to go. However, what if the connective tissue underneath an epithelial tissue had a high concentration of oxygen and the microscopically thin epithelial tissue cells need oxygen? Well, it can diffuse that different distance. So it works best over small distances. And FYI, diffusion doesn't have to occur across a membrane. It can just occur in any solution, even in a solution of extracellular fluid or intracellular fluid. And we all know that it even works well in air. If you've ever smelled a fart that wasn't yours, you know that diffusion actually worked. And so, what about surface area? If something is being diffused through a cell membrane, and for instance, in this situation, you curved the cell membrane, and you were moving it through the cell membrane, then what would happen is you would get an increased transport rate with more cell membrane. So that is what microvilli, for instance, do on the apical surface of the simple columnar epithelium that lines the small intestine. It increases the transport rate. Okay, and then if you are an ion, and we will talk about this in a lot of detail in the next set of notes, most things you can say they diffuse down their concentration gradient, but if you are an ion, you have to consider concentration and charge. So for instance, let's see in this picture, let's use this one again. Okay, so our friend potassium is a positively charged ion. And so we're just going to look at it one more time. Okay, so it's high out here, low in here. So that's its concentration gradient. Okay, so I want you to sort of try to mentally predict what potassium would do in two different situations. Potassium is a positively charged ion. If this were neutral, meaning no electrical charge, then potassium would just worry about its concentration gradient. If, however, there was a slight negative charge on the inside of the cell, and potassium is positively charged, do you think that would increase or decrease the diffusion rate into the cell? If, again, potassium was higher here, lower there in concentration, potassium is positive, the inside of the cell is negative. Is that going to increase or decrease potassium's diffusion rate? Hopefully you see that it will increase its diffusion rate because it is moving toward an opposite charge. But if the inside of the cell has a net positive charge, right, and the concentration is still high here, low there, potassium is positive, it's moving toward a positive. Will that increase or decrease the diffusion rate? Hopefully you can predict that if the inside were positive, it would decrease the diffusion rate. So when you are talking about ions, not just talking about something that's not charged, like an oxygen, a molecule of oxygen gas, then with an ion, instead of just saying that ions diffuse down their concentration gradient, ions actually diffuse down what we call the electrochemical gradient. And the electrochemical gradient is the combination of concentration and charge, electrochemical gradient. And we will talk about how to predict which direction they're going to go later on in this set of notes and then in the next one. Okay, so let's talk about our types of channels. So if when an ion can't diffuse straight through the plasma membrane, the next fastest choice is going to be a channel protein. But there are multiple types of channel proteins. And the types of channel proteins that exist are the first one, which is called a leak channel. And a leak channel kind of looks like this. It is a channel protein that just allows anything that fits to leak in and out down its concentration or electrochemical gradient. So it's basically a door that is always open. So this would be a good example of a leak channel right here. So that's a leak channel. It's just open. And FYI, channels do not have shape-specific binding sites. So if, for instance, I called this a potassium channel, it would be a usually potassium channel because anything that had the right charge and was about the right size could probably leak through that as well. So they do call them potassium channels or sodium channels or chloride channels. But since they don't have shape-specific binding sites, you can just predict what's likely to go through it because they're just kind of open. But then there is another kind of, well, three other types of channels that we're going to run into this semester. And we're going to run into these two a lot. And these two are called ligand-gated ion channels, also sometimes called chemical-gated channels. And they have two different states. Hold on, I need an animation. I usually draw this on the board, so give me just a second to look for it. So a ligand-gated ion channel. Trying to remember where it is because I don't usually do this way. Just wait right there. I know. You're busy. Okay, this will work. Okay. All right, so most of this is not going to be important to you. I just want to show channel. Come on. Okay. All right, so look at this guy right here, okay? Ignore all the rest of it. What is going to happen there is that this is a channel that has two states. This state is default closed, and then I'm going to open it. But please look, the thing that binds is not the thing that is actually going to be transported, okay? The ligand binds and opens it, and then the ion, which is sodium, is transported. If I bound and moved the thing that bound, then it would be a carrier. But it's not a carrier because it's binding and opening this ion channel. It is a ligand-gated channel, okay? And we're going to see this, of course, in detail when we get to the nervous system. Okay, so, and then the next type of channel is not anything I can draw, but kind of like the other one that we just looked at. Oh, by the way, one last thing about that ligand-gated ion channel. The ligands that open the channel are usually chemical messengers like neurotransmitters, which you just saw in that last video. It was a neurotransmitter called acetylcholine, or a hormone or a paracrine agent. So the chemical that opens the chemical gated channel or the ligand that opens the ligand gated channel is usually a chemical messenger of the type that we learned in the last set of notes. Okay, and then the other really, really common channel that we're going to see all the semester is one that is only opened when you change the electrical state of the cell. So if you like zap the cell, that's what will open it. So if the cell was sitting there at a voltage of 0 millivolts and you zapped it and it hit 55 millivolts, that's what would open it. These are not surprisingly abundant on your electrical systems, like all of your muscle tissue. All of your muscle cells have these voltage-gated channels, and your nervous system, your neurons have these voltage-gated channels. So all of the systems that you've learned multiple times that are called electrical systems, those are electrical largely due to the fact that their cells have voltage-gated ion channels on them. And we will see animations of voltage-gated ion channels in the next set of notes. And then the last type of channel we don't really run into this semester. They're kind of interesting. They're not super duper common all over the body. They're called mechanically gated channels. And these open when you stretch or squish the cell. You find them in ligaments and tendons, and they help with what we call proprioception, which means that if I wake up and it's pitch dark and my hand is near my face, how can I tell that my hand is near my face? Because I can't see my hand and I don't remember putting it there. You can tell because your tendons and ligaments are stretched in one way in certain joints and squished in other ways in certain joints. And those will actually send the signal to your brain, which interprets that as body position or proprioception. Okay, so that's simple diffusion. Simple diffusion can or cannot involve a membrane. So the membrane doesn't have to be there. But if the solute is permeable, then the membrane can be there, like with oxygen or with carbon dioxide. Okay. The more surface area you have, the better it is. So simple diffusion is straight across. When you have to use a channel, it's still called diffusion, but I call it diffusion through a channel just to distinguish it from simple or facilitated diffusion. Okay, so the next one is one that you've learned a whole bunch of times, and it's osmosis. Osmosis is just a special instance of diffusion. And what osmosis is doing is it's diffusion of water across a selectively permeable membrane. And so what water is going to do is move down water's concentration gradient. Okay, so we used to think that this was going straight through the cell membrane. When I took this, we didn't know about aquaporins. So we thought that water was small enough that it just does kind of what we think carbon dioxide is doing. This is our little Mickey Mouse carbon dioxide. No, that's not. That's water. Okay, so this is water. Looks kind of like Mickey Mouse here. So how does water diffuse? Water diffuses by osmosis, but osmosis is just water diffusing across a selectively permeable membrane. So if we get it started, so you can see water diffusing across there, but most water goes through the aquaporin. Aquaporin is a special water channel that seems to be really abundant in cell membranes. So it is passive transport because, of course, it is diffusion. It does depend upon a concentration gradient. It depends upon the concentration gradient for water. But you're almost never given the concentration gradient for water. So this kind of requires that you flip around the whole story in your head so that the concentration for water is kind of the inverse of the concentration for the solute. So most of the time you're not given this, right? You're just given that the solute concentration in the intracellular fluid is whatever, 0.3 molar or 300 milliosmoles. And the solute concentration out here is 0 milliosmoles. You don't have to do the calculations. All you have to do is figure out, okay, well, if this is the solute concentration and wherever there is not solute, there is water, what is the water concentration? Not numbers, but where is it high and where is it low? So if this 0 of solute, then it must be higher of water. 0.3 of solute, it must be lower of water. So the water concentration moves from high water concentration to low water concentration and diffuses straight into the cell via osmosis. Okay, what would happen in this cell if this occurs is that the cell would actually increase in size because the extracellular fluid was hypotonic to the intracellular fluid. Okay, and so the requirements for osmosis are a selectively permeable membrane and then two things. Water, which can cross the membrane because of all of the aquaporins, and then the solute. We would have to say this is a non-penetrating or impermeable solute because if the solute was permeable, the solute would just diffuse down its own concentration gradient. But this is a non-penetrating or impermeable solute. If you ever see non-penetrating or impermeable, know that someone is trying to get you to say osmosis, not diffusion. Okay, then let's look at this one. Okay, so again, usually you're not given the water concentration, you're just given the solute concentration, 300 to 1000. That means that the solute's low here, high here. That means the water's high here, low here. It's the inverse of what the solute is. So high water concentration to low water concentration, yes? So water is going to move out. Why doesn't the solute move? Because it's a non-penetrating or impermeable solute and it can't. So water will move down water's concentration gradient. When that occurs, water's moving out. You are going to shrink or crenate that cell because you are sitting in a hypertonic solution. Okay, so let us look at the whole isotonic, hypertonic, hypotonic situation. And let's look at this video. Did I close it? Didn't mean to. Oh, there it is. Okay. So, um, let's define the three of them first. So these solutions, what you're doing in all of these situations is you're defining the solution, which is variable, with reference to the cell, which is relatively constant. Okay, so let's think about this before we go on. Which fluid is more likely to vary in its concentration of anything? The extracellular fluid or the intracellular fluid? Hopefully you understand that the extracellular fluid is more likely to vary in its concentration of almost anything. So what is varying is the extracellular fluid, and you're holding the intracellular fluid relatively constant, at least at the initial stage of this experiment. So if I have an isotonic solution, iso means the same, that prefix, and tonos implies tension. Okay, so what I'm going to do is I'm going to have a situation. Let's see if they've got it, any of these pictures. A situation, they don't have the pictures of it. I'll show you the video in just a second. In which the solute concentration inside and outside are almost the same. When that is the case, what's going to happen is water is going to move in and out of the cell, but there's not going to be any net movement of water, meaning that this cell should not shrink dramatically. It shouldn't swell dramatically. Water is going to move about the same in and out. So that's an isotonic solution. I'll show you guys a video in just a second. Okay, so no net change in cell size. This would be like, for instance, when someone's dehydrated and you give them not straight water, you give them 0.9% saline or normal saline, because that allows their red blood cells, all their blood cells, to maintain the same size. Okay, so that's an example of that. What about a hypertonic solution? A hypertonic solution is when the solute concentration outside, remember you're always naming the solution with reference to the relatively constant cell. So this was same as the cell. Hypertonic, hyper meaning above or more than. Hypertonic solution has more solute than the cell. Okay, so this is an example right here of a hypertonic solution. More solute out here, yes, than in here. Okay, so the solute concentration is greater outside than the solute concentration is inside. So this is, for instance, why you don't drink salt water. Because if you drink salt water when you're extremely thirsty and on a desert island, what happens is that hypertonic solution, as it goes by all of the epithelial cells in your digestive tract, sucks the water out of your digestive tract. Your cells that it's coming into contact with will shrink. And of course, you are going to have a net loss in water instead of a gain in water, which is what you were trying to get to happen, because all of that water would move out of your cells into your digestive tract, and then you would have diarrhea, very little water absorption to speak of, and lose more water than you actually ingested. Okay, so that's a great example of a hypertonic solution. And with a hypertonic solution, the effect on animal cells is crenation, because the water moves straight out. Okay, and then a hypotonic solution would be this one. With a hypotonic, again, we're naming the solution with reference to the cell. Hypo means below or under. A hypotonic solution has less solute than the cell, less solute than the cell. So look at this one. So a hypotonic solution is actually going to have less solute than the cell, okay? And what's going to happen in a hypotonic solution is that water is going to move down water's concentration gradient. So the water is going to move from high water to low water. It's going to move inside the cell. And the cells will swell. And because we don't have a cell wall, because we're not a plant, the cell actually can burst. When that occurs, it's called hemolysis. When it occurs in red blood cells. It doesn't occur in plants. You can pour straight water in your plant and they'll just get nice and perky, because the plant has a cell wall and to allow it to take in all that straight water. This is also the reason that, for instance, you do not shoot up a person who is dehydrated with straight water in their veins, because the red blood cells will go through what's called hemolysis. Okay, so let's look at this. So the osmotic effect on blood cells in each of these situations.
Blood cells, like other cells, have differentially permeable membranes. As a result, they tend to be isotonic to surrounding fluids. In other words, they have the same salinity. Blood cells in normal saline solution have a slight dimple.
Let's go back just a little bit. I want you to see that this is...
Cells in normal saline solution have a slight dimple.
So this is the typical biconcave disc shape of a red blood cell that is maintained in an isotonic solution. Hypotonic is next. What do you predict is going to occur in the hypotonic solution? Is the cell going to increase in size or crenate? Okay, let's see if you're right.
When those same cells are exposed to a less saline solution, the cells begin to absorb water, growing plump.
See this? This precedes hemolysis if they kept going.
If they absorb too much water, the cells will burst.
Okay, now you should be confident in saying what the cells will do in a hypertonic solution. A hypertonic solution is when the solute has greater concentration, the solution has greater concentration of solute than the cell. So what is going to happen in a hypertonic solution? Water is going to move where? Water should move from water's high concentration inside the cell to water's low concentration outside the cell, because osmosis is just diffusion of water. Okay? And when that occurs, do you predict the cell will get larger or smaller? Let's see if you're right.
When those cells are then exposed to a more saline solution, the cells lose water, becoming wrinkled and shriveled.
And that is crenation. So when the cells become all wrinkled and shriveled. Hopefully this will help you understand. Okay, so here's what happens with people in the concept of osmosis. People tend to want to memorize that water moves toward the high solute concentration, and that while that works most of the time, it's actually not true. Water moves toward low water concentration. And sometimes the whole high solute concentration is not going to be predictive for you. So what you need to really do is be able to look at it based on the truths of diffusion. And diffusion is always that something moves down its own gradient. It doesn't move relative to the gradient of another chemical or molecule. So before we leave that concept, let's look at these two, A and B situation. This is the initial situation, and this is immediately after. So let's see if you guys can figure out what's going on with A. Again, what they're doing is they're giving you the concentration of the solute, okay? So they're giving you the concentration of the solute. So 300 whatever, it's probably milliosmoles or osmoles, milliosmoles to 150. So the solute is high here, low here. You can't move the solute because it's non-penetrating. So what I am going to do is water is going to move from where water is high. Yeah, it's higher out here than it is in there. Water moved in, yes, and the cell got bigger. So with this, this is, of course, osmosis because water is moving across the membrane. But what kind of solution was this originally? What was this? Was this an isotonic solution? Was this a hypotonic solution or a hypertonic solution? By looking at the outcome. So water moved from high water concentration to low water concentration, okay? Because the solute was lower out here and higher in here. And so the water concentration was the inverse of that. So this was a hypotonic solution. Okay, so let's look at this one. So what's going to happen here is, again, you're giving the solute concentration, but that's not what's important. What's important is the water concentration. So 600 solute outside to 300 inside, so high solute, low solute. That means higher water concentration here, lower water concentration here. Water is going to move out, okay? Cell smaller. So this was a what solution? Isotonic, hypertonic, hypotonic solution. This was a hypertonic solution. Okay, so Um, so please practice this because it's going to be really important when we get to the kidneys. And if you had trouble with it, go ahead and look back at and listen to my lecture from last semester as well. And let's just watch it one more time. So this is, although it looks like typical diffusion, what it is osmosis because it's water diffusing across a selectively permeable membrane. All right. And then the last thing that I'm going to do today is just what we call the mediated transport systems. And I'm going to stop after I finish facilitated diffusion because doing primary active transport, I really want to do that with you in front of me. So a mediated transport system is going to be when you use not a channel, this is not mediated transport, but a carrier, okay? And the fact that it's a carrier matters because carriers have shape-specific binding sites. They're going to be slower than channels, and they're going to be super duper specific about what they move, okay? Shape-specific binding site like an enzyme. Okay, so a mediated transport system is going to utilize a carrier protein. It's going to bind to a specific substance and help it across the membrane by changing shape. Since it's a shape-specific binding site, don't be at all surprised that you can turn these on and off, just like you can with an enzyme. Remember, you could bind a modulator right here, and it would temporarily change the shape. You can do the same thing with these. For instance, maybe this glucose carrier does not fit glucose unless insulin binds right there. So it's subject to regulation. And the other thing is if you're shape specific, you're subject to saturation because like an enzyme, it can get full. So please look at this and notice how similar this looks to the whole enzyme thing. With the enzyme, what you had is the concentration of substrate and the reaction rate on the x-axis and the y-axis. And what you saw is, of course, the reaction rate. As you increase the substrate concentration, the reaction rate goes up until you hit saturation. Well, saturation was all about having a shape-specific binding site that could be full. But this says as the concentration gradient increases, the net flux or the rate at which the glucose, for instance, was moved across the cell membrane. This says as concentration gradient goes up, the net flux goes up until, of course, your carriers are full. When your carriers are full, it doesn't matter if there's a greater concentration. You're not going to get any more net flux. This will matter very much when we get to talking about diabetes and why, for instance, type 1 diabetes will cause you to pee like crazy. What it is you take in sugar. This is a short version. We'll get it in detail later. And since you take in a whole bunch of sugar and it's still in your extracellular fluid or your blood plasma, a whole bunch of it gets pushed out into the urine. It's called filtrate at that point, in the kidneys. Okay, and that just happens by filtration. Okay, but the reabsorption mechanism is based on a carrier protein. So it's subject to saturation. So you try your hardest to bring all of the glucose back in, and you do really well until all of your glucose carriers in your kidneys are full or saturated, and then you can't bring any more back in, and you just end up peeing out the rest of it. Okay, so subject to saturation limits, and that matters. Okay, so let's just talk about facilitated diffusion, and then we'll be done for today. So facilitated diffusion is simply diffusion through a carrier. One more time, the only reason that you would do that is because you're not lipid soluble like oxygen, so you can't do simple diffusion. You're not small enough to diffuse through a channel. So what you are going to have to do is plan C, which is diffuse through a carrier, which is going to be slower than diffusion of either of those other two ways. So diffusion through the carrier or facilitated diffusion is going to be slower than simple diffusion or diffusion through a channel. But still, it doesn't require ATP. It's just limited in its locations that it can diffuse. And also, it has to wait until it binds, dumps, binds again, dumps. So it's going to be slower. So, but it doesn't require ATP. It only moves things from high concentration to low concentration. And so the net movement is going to be from where glucose is high to where it's low. So we would know that this picture is facilitated diffusion if I just told you that glucose had a high concentration here and a low concentration here. Then this would be facilitated diffusion of glucose, okay? If I don't tell you the concentrations, then you can't quite tell me that. Okay, so an example, amino acids move from, for instance, the blood plasma into the cells through a specific carrier because, one more time, they are not lipid soluble. They can't diffuse straight in. They're too big to diffuse through a channel. So let's pretend that this was an amino acid transporter this time. Amino acids are mid-sized, back to this again. Not huge, like you could count all of the atoms in them relatively easily. Mid-sized and water soluble. So how did they get across the cell membrane? They have to move through a carrier, okay? And that is facilitated diffusion. It's also the really common way of getting sugars inside the cell. Okay, so what we will do on Wednesday is pick up there and talk about primary and secondary active transport. But there are questions about the stuff that I just covered on your quiz, and I don't have time to cover this again in class because we couldn't afford to miss that day, much less for me to take another day. So I want you to watch this as many times as you need to. Again, talk to your friends, your students in your class or the other class to get through this stuff because this is stuff that you've had before. Really, the different spin on it that I'm trying to enforce to you is that I need you to get to the point that you're comfortable predicting how things are going to be transported across the cell membrane. The next thing that we are going to do is try to transport them against their concentration gradient. And when we do that, so if for instance sodium was low here, high here, and I wanted it to go that direction, I would have to pump it. Same thing with potassium. Potassium diffuses in just fine by itself, but what if I wanted potassium to go against its concentration gradient? Then I would actually have to pump it. So, oh, that's funny that they had it diffusing that way. Because potassium is, just FYI, not particularly important. Potassium is generally a higher concentration in the cell than out of the cell, so that's a strange little example that they used right there. Either way, the visual will still work for you because you don't have to memorize that yet. Okay, so have a good snow day, be careful and be safe, and I will see you guys on Wednesday. Keep working on your quiz. It's not due until I think after class on Wednesday. I can't quite remember, but not today anyway. I will see you Wednesday.